let's start with the very trivial thing, maybe, but it's the definition of what is the sequencing. So it is the process of determining the primary structure of given by a polymer to know the order in which the 4 nucleotide bases occur within a nucleic acid molecule. Then sequencing technologies are continuously improving, and in literature they are referred as different generations based on the main technology used. So there are several characteristics that are characterizing each of the generation. So first generation is the one that only one read is obtained at a time, and it was the method developed in 1977 by Sanger, and the second generation, which is also referred very often as the next generation sequencing, has this characteristic that a sample is amplified by PCR step and many reads are obtained simultaneously. And the third generation is characterized by single molecule real-time sequencing, and during which long reads are obtained. So we will go a little bit in detail about all of these. So let's start with the Sanger sequencing. It's also known as a chain termination method or sequencing by synthesis. So as I mentioned before, it was first developed in 1977 by Sanger from Cambridge University, when he was afterwards awarded with Nobel Prize in Chemistry in 1980. So chain termination PCR works just like standard PCR, but with one major difference, that there is the addition of modified nucleotides, so called the B-deoxyribonucleotides, and compared to the deoxynucleotides, they have they lack the oxygen atom, and hence they cannot form a link with the next nucleotide in the chain, and this chain is terminated therefore. So the library prep starts with the denaturation of the double-stranded DNA using heat, and then the primer can attach to the sample, and the third step is that the sample is then added to the 4 reaction solutions that contain already this PCR mix. Then PCR products are purified, and double-stranded DNA molecule is denatured with heat and fixed in formamide. So basically we have PCR reactions occurring in 4 different tubes, which contain differently labeled 4 different nucleotides. So afterwards, when we want to visualize our results, we use gel electrophoresis, and our fragments would be separated by size on a gel, and therefore we could see the different nucleotides that form our sequence. The next is Maxim Gilbert sequencing, which also is part of first generation. It's also known as the chemical degradation method, and it starts with phosphatase cleaving of this 5' phosphate group, and instead polynucleotide kinase is adding the radioactive phosphorus instead. Then the DNA is denatured with the metal sulfoxide at 90 degrees, and then we have the similar like before four chemical cleavage reactions. So using different combinations of these chemicals, we can cleave the target DNA in specific places. So, for example, in purines, you would use the dimethyl sulfate, and in pyrimidine, to cleave pyrimidines would be hydrazine. And and in this picture, you can see the specific chemicals that are added to each of these reaction too. Next is similar like before visualization step, and it's also done like it's also electrophoresis and on acrylamide gel. So similar like before, we can then therefore conclude our final sequence by just looking at these different sized fragments that we obtained. This is small comparison between these two methods. So in case of Sanger sequencing, we use fluorescent dye for labeling. In case of Maxim Gilbert, we use radioactive labels, and the average length of sequence is a little bit different. Then the plus of these both methods is the high accuracy level, but unfortunately they both are slow and time consuming, and a large amount of DNA is necessary. Also, due to toxicity of the reagents, Maxim Gilbert method is not very widely used, and Sanger sequencing technology remains very useful for applications where the high throughput is not required, or when it's necessary to fill the gaps in the sequencing data, or in difficult to sequence areas, for example. And is still used as a golden standard for validating new sequencing approaches. So next is second generation sequencing. So in this group, there are different technologies. However, there is also common characteristics in these technologies. So in second generation sequencing, the many million reads can be obtained in parallel. Therefore, there is higher throughput, and there is higher speed and moderate cost, as well as the sequencing output is directly detected without the need of electrophoresis, but there's need of optical system, and the sample has to be amplified by PCR. Mainly two types are used, it's bridge amplification and emulsion PCR. So so this is the common workflow as well that all these methods share. So the first step is library preparation, then is the sequencing step, and then is data analysis. And library preparation is also the first step. So the library preparation are pretty similar among all of these technologies. So library preparation first steps would be the fragmentation of our DNA. That can be done by enzymatic or physical methods. Then after that, we would do end repair, which would make blunt ends of the previously fragmented DNA, because during the fragmentation it can be a little bit damaged, or there can be some breakage in one of the strands. So after is the A-tailing or adenylation, during which the adenine tail is added to the strands, and it's also done to ensure that in the next step, the adapter ligation will occur properly because adapters have the T-tail usually, so they would attach to the adenine tail. So next step, as I say, is adapter ligation, and adapters are basically sequences that are already known before. So we add adapters for several reasons. Adapters contain the sequences that are complementary to the sequencing primer, so they will serve the function of site where the sequencing primer is attaching. And also they serve the identification for different samples, since we can sequence many samples at the same time. And after sequencing, we will gain only sequences. We will need to know which sequence belongs to which sample. So adapter sequence contain also very short index or barcode that will serve as ID for each sample. And the next step is the amplification step. And as previously mentioned, this can be a little bit different depending on the technology. So next, so sequencing step between these different sequencing methods is more various, and we will go a bit in detail. So first is Illumina. Illumina technology is sequencing by synthesis approach and is currently the most used technology in the NGS market, and still is the most used. So after the adapter is attached to the sample, this DNA is transferred to the flow cell. And the flow cell is a pattern microarray that contains billions of nanowells, and each nanowell contains oligos that are complementary to the adapter sequence. Currently, there are two kinds of flow cells. There's random and there's patterned. In random flow cell didn't have the nanowells, whereas pattern have, and therefore. In random flow cell, overclustering could occur since the DNA could be attached to oligos and flow cell unevenly and be unevenly distributed. So in patterned flow cell, this is avoided due to the fact that microwells are in constant distance from each other. So they're less susceptible to overloading and more tolerant to broader range of library densities. So after our DNA is attached to the flow cell, the polymerase and synthesizes the complementary strain of the sample. Then the double-stranded molecule is denatured. The original strand is washed away. Then this newly synthesized strain folds and attaches with the other end to the other type of oligo on the flow cell. And polymerase again makes the complementary strain. So during this step, the molecule is creating this bridge composition, and therefore it's also called bridge amplification. So yeah, this process happens simultaneously in millions of clusters on the flow cell. After that, the reverse strands are washed away and sequencing can start. The sequencing consists of 3 stages. So it's incorporation stage, during which 4 different nucleotides that each have their own specific fluorescent dye are flooded over the flow cell. And then one of the complementary nucleotides is incorporated in the chain. And the next step is imaging phase, during which this emission wavelength and signal intensity that is determining the base call, which base is called. And the third step is the cleavage of the terminator and cleavage of the fluorophore, and then the process can repeat as many cycles as long the read that we need to obtain. So yeah, there are wide variety of the sequencing devices. The overall chemistry is very similar, but some of these use 4-channel sequencing chemistry like HiSeq and MiSeq, and some use 2-channel like NextSeq and MiniSeq and NovaSeq. Yeah. So the main difference is data output and read number. Then the next is semiconductor sequencing, the ion torrent. The ion torrent is basically an ultrasensitive pH meter that detects hydrogen ions released when nucleotides are incorporated during DNA synthesis. So this process eliminates the need for optical system, thereby simplifying the overall process. But the technology is still a wash and scan system, like all current second-generation technologies, requiring PCR amplification of the DNA in each well, as well as termination events that typically halt the sequencing after each nucleotide is incorporated. Yeah, also no modified nucleotides are necessary. So the library prep, during library prep, the DNA sample is fragmented and adapters are added as all the previous. Then the sample is added to the water and oil emulsion, which obtains beads that are covered with oligos that are complementary to the adapters. Then each bead is emulsified in water in oil droplet together with the PCR reagents. And ideally, one droplet contains one bead, PCR mix, and a strand of DNA. And these droplets act like microconductors that for PCR to occur and amplify our DNA strand. So first, the strand is elongated with DNA polymerase by adding DNTPs. Then the double strand is denatured, allowing for the strand to ligate to another side of the surface of the bead. And eventually, approximately 1 million copies of the target is amplified on the surface of the bead. So basically, then our beads are ready to be loaded on the chip. The chip, so each of the chip contains a top layer covered in tiny wells, and each well is just large enough to fit one single bead. And sequencing primers and DNA polymerase are bound to the templates and pipetted into a chip's loading port. Then the chip is spinned to ensure that the beads are spread evenly across the chip, and and the size of the chip is determined by the number of bases needing to be sequenced. There are different kinds of chips. For example, here is the example of 5 different types of GeneStudio S5 system, which is not so new anymore, but it's here, as an example. So, okay, and then beneath the microwell is a sensitive layer, and below is ESFET sensor plate, which is ion sensitive field effect transistor, and it's used for measuring ion concentrations in solution. Then when we have loaded our sample on the chip, we can proceed with sequencing process, and during the sequencing process, each nucleotide is being introduced onto the chip at a time. If a nucleotide binds to the DNA strand, then the hydrogen atom is released and the pH of microwell is dropping. Chip transfers this chemical information into electrical signal. and captures the voltage measurements from it in the base, and the base is called. The unattached DNTP molecules are washed out before the next cycle, and a different DNTP is introduced. If there is more than one nucleotide of the same kind, if we have to sequence homopolymer, for example, then the several hydrogen atoms are released and a stronger signal is detected. However, it can be problematic to detect how many bases were added if the homopolymer is longer than 7-8 bases. The next one, okay, sorry, there's, for example, 2 of the ion torrent devices, and yeah, so it is improving also its capacity and allows with each of the next of the device allowing to sequence larger amounts, obtain larger amounts of data. So the next is MGI DNBSEQ technology, and the difference from the previous is that instead of using the standard PCR to amplify the sample, rolling circle amplification is used. And yeah, so the library prep after DNA is fragmented, adapters are added. Then we need to circularize our molecule, and that is done during this circularization process right here. And during this process, the split oligo that is complementary to both 5' and 3' ends of the adapters will hybridize and make a circular molecule. After that, we need to amplify the circular molecule during DNB making process, and DNB stands for DNA nanoball. DNA nanoball is generated through this rolling circle amplification step. So what happens during this step is that high fidelity polymerase is used and it's basically amplifying the initial circular molecule multiple times. And the advantage is that it's replicating the original template and not the copy of the copy, as for example in bridge PCR, therefore reducing the risk of introducing errors in this process. So after that, we have made our DNB nanoball, and it's ready for being loaded on the chip. So the chip surface is covered with the binding spots that have positive charge at a constant distance, allowing only one nanoball attached to it, and therefore ensures high chip utilization, and it's making the detection of optical signal easier to distinguish. So after that, the sequencing can start, and it's basically sequencing by synthesis. So again, it's the same 3 phases, which is incorporation phase, imaging phase, and cleavage phase. So similarly, like in Illumina sequencing, incorporation phase, complementary nucleotide is attaching to the template. Imaging phase, the optical system is detecting the light emission of the fluorophore, and cleavage phase, the block is removed, the fluorophore is cleaved off, and the next nucleotide can be incorporated. And more about this technology we'll probably hear in the video after this lecture. Yeah, and here are some of the sequencers offered by DNBSEQ. So depending on the needs, of course, different sequencers are more suitable for several different applications. So DNBSEQ 400, for example, allows us to sequence on one flow cell on one run for full human genomes. But for example, the DNBSEQ T7 has much higher capacity, so it's even it's suitable, for example, for population genomics projects. And also you can, this is parameters for one flow cell, and simultaneously 4 flow cells can be sequenced. So the next is third generation sequencing. So third generation sequencing is sequencing DNA molecules without amplifying them through PCR, therefore overcoming the issues related to this PCR step, so there's no PCR bias. So also they allow to obtain much longer reads than second generation sequencing. Okay, so this is not true. It's less expensive because this is the cost of the third generation sequencing is still higher than that one of the second generation, and also as a con would be that it's usually requiring to use fresh materials or intact cells. That's not always possible. Also, there can be the data analysis can be a little bit more challenging due to the fact that the database systems for interpreting the complicated structural variants are rare, and also standard molecular biology methods are optimized for short read sequencing. So also this for obtaining longer fragments for this long read sequencing can be also probable that the optimization of the methods still needs to be done. And also a pretty big drawback is the accuracy. It's less accurate than second generation, so the error rate is 10 to 15%. The difference is that, again, these errors are more random. They're not related to the PCR bias. And these are the two examples of most used third generation sequencing techniques. So one is Pacific Biosciences and the other is Oxford Nanopore. So Pacific Biosciences, single molecular real-time sequencing is one of the most commonly used third generation sequencing technology, and compared with previous two generations, so the biggest advantage is this long read sequencing that requires no PCR amplification. So library is being constructed by fragmenting, size selecting, and attaching the adapters, similarly like in previous generation. In the end of this template called SmartBell is created. It's a closed single-stranded circular DNA, which is created by ligating hairpin adapters to both ends of target double-stranded DNA. Then this SmartBell is anchored to the bottom of glass surface of the zero-mode waveguide. Zero-mode waveguide is a sub-wavelength optical nanostructure fabricated in a thin metallic film, and it allows individual molecules to be isolated for optical analysis at physiologically relevant concentration of fluorescently labeled biomolecules. So 4 types of nucleotides are labeled with different fluorescent dyes, similarly like in previous technologies. And as a nucleotide is held in the detection volume, by the polymerase. Laser light travels through the bottom surface of the zero-mode waveguide and not completely penetrates it. So it doesn't completely penetrate it because the zero-mode waveguide dimensions are smaller than the wavelength of the light. So therefore, it allows selective excitation and identification of light emitted from nucleotides. And the base can be called. So then the next nucleotide binds to the template in the active site of the polymerase, initiates the next fluorescence pulse, which then corresponds to the next base. Then we have Oxford nanopore. The significant advantages of the nanopores include the label-free, ultra-long reads, high throughput, and low material requirement. Each of these greatly simplifies the experimental process and can be easily used for DNA sequencing applications. So using nanopore sequencing, a single molecule of DNA can be sequenced without the need of the PCR amplification or chemically labeling the sample. A nanopore is simply a small hole of the order of 1 nanometer in internal diameter that is made up of certain transmembrane cellular proteins. The nanopores are inserted into a synthetic polymer membrane that has very high electrical resistance. The single-stranded DNA is introduced into the nanopore by entering from the microscopic opening, and this DNA sample is then introduced in an insulating membrane between two compartments, and is filled with saline solution, and an electric potential is applied across it, so across the membrane. And the way in which the bases influence the electric field through the nanopore is measured. So these measured data can be decoded to retrieve the DNA sequence. So yeah, so as I said before, then this electrophoretic force is induced, and by positive electric field, which is applied, and it attracts negatively charged DNA and drags it in the pore. So, and then the difference in resistances of different bases allows the DNA sequence to be determined. And base calling is taking place in the real time. And here are some of the Oxford nanopore sequencing devices. Okay, and this is a little comparison between these two third-generation sequencing methods. So PacBio is using fluorescent signals to detect nucleotides, and Oxford nanopore is using electrical signals caused by nucleotide blocking the pore. The common thing is that the polymerase and the strand is fixed, and the polymerase is is fixed in the spot while the strand is moving through. So yeah, every sequencing generation and platform, by reason of its methodological approach, carries characteristic advantages and disadvantages, which then determine the fitness for certain applications. And more about the applications. So, so this is like a little summary of the applications of next generation sequencing, or just sequencing. There can be transcriptome, exome, metagenomics. As in previous presentation, we saw what what can be the usage of the data gained from metagenomics, single cell genomics, epigenetics, and so forth. So DNA sequencing mostly can be divided into two categories, can be de novo and resequencing. And in the case of de novo, reads are obtained from an unknown sequence and either assembled to reconstruct the sequence or compared directly to reads from other unknown sequences. In the case of resequencing, reads are mapped or aligned to known reference sequence. So whole genome sequencing, for example, is the most comprehensive method for analyzing the genome, because this method entails sequencing all of an organism's chromosomes, and as well as DNA contained in mitochondria, and for plants also in chloroplasts. So target sequencing, now exome sequencing would be a genomic technique for sequencing all of the protein coding regions. So basically it's 1% of the size of the whole genome. So in this case, if we want to look only at the coding part, we would save the resources by just sequencing that part. Then is the target sequencing. A subset of genes or regions of the genome are isolated and probably also enriched and sequenced, and this also allows us to focus on the time and expenses. Cancer sequencing provides more information in less time compared to traditional single gene and array-based approaches. And metagenomics is study of genetic material recovered directly from environment. So it's usually used when we are studying microbial communities where we can't separate one microbe from another. Then some examples of RNA sequencing. The total RNA sequencing detects coding plus multiple forms of non-coding RNA. This method can be used to examine regulatory regions, expression levels or transcripts, splicing patterns, and to identify exons and introns and their boundaries. And RNA exome capture sequencing can be used as a combination of RNA-seq with exome enrichment. And this method captures only the coding regions of transcriptome, allowing higher throughput and requires lower sequencing depth than non-exome capture methods. Interesting application is single cell sequencing that examines the genomes of transcriptomes of individual cells. So they enhance, these assays enhance the study of cell function and heterogeneity in time-depending processes such as differentiation, proliferation, tumorigenesis, and plays particularly important role in understanding cancer and phenotyping tumors, because as we know that even within the same type of cancer, the cells can be very various. Another interesting approach is bisulfite sequencing. That is method that's used for research in epigenetics. And it's based on the fact that DNA methylation is an epigenetic mark and is implicated in repression of transcriptional activity. And after the DNA is treated with disulfide, it converts unmethylated cytosine into uracil. However, the methylated cytosine remains unchanged. In this way, we can discover, for example, silenced tumor suppressor genes, and so on. So the challenges, the next is the challenges. Yeah, so these are pretty much the challenges that we still have that are related to sequencing, sequencing. So first is still data storage, because of the sheer amount of data being generated, during sequencing process is enormous. And yeah, it's like the BAM file of single human genome, 30 times coverage would be 90 gigabytes. So a relative modest project of 100 samples would generate 9 terabytes of BAM files, for example. So also it's very often necessary to save the data from the same sample in different stages of data analysis that just can double or triple the amounts in storage that we need for the same sample. Security is also still an issue because it's important to maintain the privacy and confidentiality in data management practice. Yeah, so and data analysis is still the bottleneck. It's also related to the first challenge, the amount of data that is produced, and also the speed at which it is being analyzed. So the data analysis is still quite time-consuming process, even though we are using automated pipelines. And yeah, and also the challenge is to create new analysis pipelines and to appropriately select the right tools from the many existing ones. So different, also different DNA data analysis tools have different biases and abilities to covariance. And especially in clinical environment, it's important to use a united standard for interpretation of different variants. Future, okay, yes, this is about maybe future generations. So actually it's already method that is already used. Yeah, so maybe we will switch in the future more to this fourth generation method that is basically offering us to do the sequencing in situ. That basically means that we do sequencing directly in the cells and tissues. And yeah, and so yeah, so nucleic acid sequencing has undergone rapid and impressive development in just a few decades. So yeah, and it also reflected in the way that medicine is changing the genomic era, in which the sequencing each person's genome might be a real possibility for routine clinical practice. Yeah, that's it for me. Thank you. I'm a little bit ahead of the time, so maybe there are some questions or yeah.
Yeah, thank you, Alise. A very wide and still very good presentation. If there are any questions, please ask. I have a question. What are the most like not mistakes, but what are the most popular or often reasons when you know that sequencing run is not successful? What are what could be reasons for that?
Well, probably number one reason would be low quality sample, but okay, in theory, that shouldn't happen if you are actually doing everything right and you did the quality check just before sequencing. If there are some problems, you should not continue with sequencing. However, sometimes this is the case still, and yeah, and then it can also, of course, be some very random mistakes of the hardware. Sometimes that happens. It's quite rare, but it happens. In this case, of course, sequencing run must be repeated. Yeah, but the main reason would be the mistakes made during the library prep. So it's still the same rule, garbage in, garbage out. So yeah, it can be many different